Ik dacht, je heeft wel even
I hereby open the meeting. Mr. Schiewe, I bid you welcome. You are appearing before a committee appointed by the Doctoral Board of Erasmus University Rotterdam to defend your thesis and the appended propositions. But before we start the discussion, I invite you to give a short introduction in which you tell us about the subject and the aims of your study and your findings. The floor is yours.
Thank you, Mr. Rector. Good afternoon, friends, colleagues, and esteemed professors. I am here today to expose the work of my PhD. And during my PhD, I worked in the laboratory of Dr. Ricardo Fodde, who works on stem cells and cancer. And specifically, I focused on colon cancer. There are four main reasons of why we do get colon cancer: age, diet, inflammation, and genetics. And we believe that these four etiological factors act on the stem cell niche, altering it. Colon cancer has a very well-defined sequence of mutations through which it progresses. And we believe that the four etiological factors just mentioned act upstream these mutations. And as I said before, that they alter the stem cell niche. During my PhD work, I mainly focused on two out of the four factors: inflammation and genetics. But before introducing my work, I would like to first introduce the organ that I study, which is the gut. The gut is divided mainly into two segments, the small intestine and the large intestine. And you can see the small intestine on the left with these digitiform finger-like protrusions, where adjacent to them there are crypts. And it differs from the large intestine because in the large intestine there are no finger-like protrusions. And it's in the pit that you can see next to these fingers where we find the stem cell niche. When we look closely to these pits, we will see that there are mainly two types of cells residing at the bottom of these pits. One is a bulkier granulated cell, which is called Panna cell. And what this cell does, it is a niche cell. And it's a niche to the cells next to it, which are more slender cells, columnar shaped, and they are LGA5 stem cells. And what do stem cells do? So every day, our gut loses millions of cells, and all of these cells need to be replaced. And that's the work that stem cells have to do. And Panna cells help them in their job by being their niche. Panna cells were indeed the main focus of my studies during my PhD, and they are historically known to secrete antimicrobial factors and to keep the bacterial flora in check. They provide, as I already said, niche support to the LGA5 stem cells, They underlie de novo crypt formation during development. And as well, they respond to environmental factors by modulating the niche support depending on which environmental factor they're exposed to. And the tool that we use to study the gut was invented by the Clevers lab, and it's the organoids. And what are organoids? Organoids are a way that we have to grow organs in a dish. It's like if I take a piece of your gut and I can literally grow it in a dish in the lab, and this gut behaves like our guts behave every day. They grow and shed cells. And in our lab, we use a modified version of the organoid tool where we can take the single cells from the crypts and mix stem cells from mouse A with panel cells from mouse B and address the function of either the stem cells or panel cells from one or the other mouse. During my PhD, I had mainly three questions. First one, where do intestinal stem cells and panel cells get their energy from? Second one, what is the role of panel cells as niche cells upon inflammation? And last but not least, is there a role for panel cells as stem cells in inflammation and cancer? I will first address the first aim. And what we did in collaboration with the Burgering lab was that we isolated cells from the mouse and we checked how they took their energy from. And just like it happens with cars have two ways to get their engine going. Either they use gasoline or they use diesels. And with cells, it's more or less the same, meaning that they can use either oxidative phosphorylation or glycolysis. And what we observed is that indeed stem cells in the gut use oxidative phosphorylation, whereas panel cells use glycolysis. But furthermore, we saw that there was a metabolic teamwork. We observed that these panel cells were stimulating, boosting the metabolism of the LGF5 cells by secreted metabolites. Then the second aim of my PhD was, what is the role of panel cells as niche cells upon inflammation? And to thoroughly address that, I need to go a little bit back in history. It was 1993, I was starting my elementary school when the group of Bill Dove first was working on the APC mean mouse, and they saw that this mouse was getting 90 tumors per intestine, but when they crossed it with a red mouse, they observed a striking reduction in tumor multiplicity. And you can basically do the trick again and again, crossing the white mouse into the red mouse, until you have an APC mutated mouse with almost no tumors. So this was pointing towards a gene in the red mouse diminishing the tumor multiplicity. And indeed, that was the concept of the genetic modifier of APC, and later on with new genetic tools, it was identified that the genetic modifier was the secretory phospholipase A2. So what are phospholipases? Phospholipases are molecules that promote inflammation, and they are mainly deputed to produce the main inflammatory molecule, which is prostaglandin E2. And the interesting thing is that they're expressed only in panel cells. So therefore, what we elucidated is that in normal conditions, panel cells have phospholipase intracellularly, inside their cells, and this inhibits the niche function. However, in inflammation, phospholipases are highly secreted, and they promote the niche function. So there is a dichotomy where in normal condition, phospholipases have an inhibitory effect on the niche, but in inflammation, phospholipases have a positive effect on the niche. And interestingly, what we observed is that when these phospholipases were mutated, so completely absent, the mouse was susceptible to colon tumors. And that is why phospholipases are genetic modifiers. Last but not least, is there a role for Paneth cells as stem cells in inflammation and cancer? We observed that by inflaming a mouse, stem cells do not survive. And this is striking because I just told you that stem cells are deputed to renew the gut. Their job is to make new cells every day, but in inflammation, they die. That's counterintuitive. And what we noticed is that Paneth cells were actually doing the job. They were de-differentiating and creating new cells and regenerating the gut. And we saw that this happens via a secreted factor called SCF. So, in conclusion, I would like to say that the dogma is that a stem cell can only differentiate, and it never goes back from its differentiated state. However, what we found is the contrary. Paneth cells can do the trick. They can go back, de-differentiate, and regenerate the gut. And last but not least, we also saw that all this regeneration comes with a cost. And the cost is the increase in tumor susceptibility. And with this, I would like to conclude and give back the word to the rector.
Mr. Candidate, I thank you for your presentation. I call now upon your promoter, Professor Vodde, to start the discussion. Professor Vodde.
Thank you, Rector Magnificus. I will not ask any question to my own candidate, and I will give the word right away to the first opponent. Thank you.
And the first opponent is Professor Katajisto. Professor Katajisto is Professor of Aging Biology at the University of Helsinki. Professor Katajisto, on behalf of the Erasmus University, I thank you for your participation in the opposition, and the floor is yours.
Thank you so much. So let me start by congratulating the candidate on an amazing body of work, something, of course, that is very interesting to me and our laboratory as well. So one of the themes that are going through all of the work in your thesis is this reciprocal interactions between two cell types, the Paneth cell and the neighboring LGR5 expressing stem cells. And you use a very clever system, the reconstitution assay of organoids, quite often to tease out dynamics between these two cell types. And let me start by probing you a little bit on the possible caveats that you see in this approach. Can you think of any?
Highly learned opponent, thank you for your question. The possible caveats of this approach are that these cells need to be single-cell digested and FACS sorted, and this might already change part of their biology and also alter the viability of the cells, which might not act anymore as they would act in normal conditions. Another caveat that I see is the absence of stromal factors. Now we are looking only into the epithelium, but these epithelial cells are usually surrounded by stromal cells, which will also alter their biology.
Very good. And when you isolate these cells and put them back into the culture together, what needs to take place? What do you actually score, and what do you see as a meaning of an organoid growth when two cells meet?
When two cells meet, I first count the clonogenicity, meaning how many cells form a lumen, and secondarily, I count the number of buddings. And the buddings are cryptomains formed by these organoids, and they indicate regenerative capacity of the organoid.
And you use this assay widely through your work, as I mentioned, to probe on cellular or cell type specific reactions. So if this connection between the two cells is very important, and like you indicate, for example, in your work with the phospholipases, that the connections or the interactions, even in these movies, seem to be delayed. Is that what you're actually assaying here? Just cells either connecting or not connecting in the assay, or what is the actual readout in the end, the organoid formation about?
Well, I believe that the formation of the doublet, LGR5 and Paneth cells, in our case, it's not a major issue, because we are centrifuging these cells before plating them. As such, doublet formation occurs in the tube, and then the cells are plated. However, there is also the possibility that even when centrifuged, altered Paneth cells might not bind to LGR5 cells.
And probing you still on the maybe, if I still have a minute of time, or am I out? Yep. On the actual findings from this assay. So, for example, the ability of single cells instead of cell pairs, only even Paneth cells are capable of forming organoids after DSS treatments of mice in your work. What happens there? How does the organoid form? There's no connections between two cells in there. What are we looking at then? What does this tell you?
We did movies also of organoids growing from Paneth cells. And what we observed is that one Paneth cell alone never forms an organoid. It always needs at least another Paneth cell to then form an organoid. And what I believe is happening is actually a proof of the lateral inhibition. One Paneth cell will be high in Notch and induce the de-differentiation of another Paneth cell.
And this is leading to my final question. Okay. In the DSS model, indeed, you lose the stem cells from the mice. Do you think that is preconditioning the Paneth cells, somehow subclassifying them already in vivo, or via lateral inhibition, and that is what you are looking at in the co-culture model, or what is the mechanism really about?
I believe that there is a preconditioning in vivo. First, right now, I believe that we don't have the tools to really define different maturation stages of mature Paneth cells. In my opinion, a Paneth cell that is residing, for example, 80 days in a crypt will not de-differentiate, but a younger Paneth cell, which exhibits all the markers of mature Paneth cells, probably will. Moreover, the ones that will receive SCF will then be more receptive to this de-differentiation.
Thank you very much.
I thank you, Professor Katajisto, for your opposition, and the opposition will be continued by Professor Burgering. Professor Burgering is Professor of Signal Transduction at Utrecht University, and I thank you for your participation in the discussion. Professor Burgering, the floor is yours.
Thank you, meneer de rector. Dear candidate, I will not start but first congratulations with the completion of a very, very good thesis, and actually an excellent thesis. And besides these compliments, I also want to thank you for the nice collaboration we did with Maria in our lab, which also gave a very good result, so to speak. But I will not leave here in Rotterdam only compliments. I will also like to ask some questions. And the questions are mainly focusing on chapter 5. I think I will start firstly with some technical questions. So the model here is that DSS treatment will result in the loss of stem cells, and then Paneth cells through upregulation of c-KIT will require stem cell function. And one of the means by which you test that is by using this organoid assay just discussed. And then I tried to go through all the figures, but in all these figures you use Paneth cells from DSS-treated mice, but you never do that minus and plus KIT ligand. So how do you explain that these Paneth cells taken straight from the DSS-treated mice already give an increased reconstitution of organoids without ligand?
Highly learned opponent, thank you for your question. I believe that the reason of the increased clonogenicity of Paneth cells under inflammation is because inflammatory cells already express and secrete the SCF. Furthermore, via RNA-scope in chapter 5, we show that even epithelial cells secrete a lot of
I
SCF.
may interrupt. So now you're saying that the ligand also comes from the Paneth cells, because I couldn't find that in your
The ligand
or the KIT receptor.
is given to the Paneth cells by inflammatory cells that infiltrate the gut.
But they're not present in your organoids assay.
They're not present in the organoid assay, so they are in vivo given by the inflammatory cells and by other cells, and then we isolate these Paneth cells who already had received the SCF.
Ah, bon, okay. And then you go on to show that by using inhibitors that it's the PI3 kinase pathway that's relevant downstream of KIT. And you make a remark, and I forgot about it, but then in your introduction you show the paper specifically addressing the role of mTOR in Paneth cells. But your statement is that rapamycin has no effect at all in your assays. How do you explain that?
So rapamycin had no effect on organoid clonogenicity, but there were morphological effects.
And the morphological effects then indicated differentiation, or?
No, the buddings were elongated, which might be suggesting that there were more transit amplifying cells or other kinds of more differentiated cells, but not Paneth cell dedifferentiation.
Okay, so at least you come to my favorite pathway, the AKT, although we call it PKB, but that left aside. And then you do a couple of experiments, and with all due respect, I'm not convinced. So, for instance, if you do a Western blot and you have such a weak signal of the phosphorylated AKT, why didn't you include a positive control? Because, you know, how can any reader, or maybe not that knowledgeable, see what the biological meaning of these small differences are? So, you know, a positive control would at least indicate what a real induction is. How do you think about that?
A very learned, highly learned opponent. Thank you. I do think a positive control is always useful to be included, of course. However, at the moment when the experiment was designed, I did not think about it.
Yeah. Well, that happens to a lot. So, but you know, the reason why I want to drag on this is that then I go to figure 6, and then you do again a Western blotting, but now with GSK3. And then I became puzzled, because the LGR5 cells are the stem cells. They have high Wnt signaling, and so GSK3 should be inhibited. But I see no sign whatever of inhibitory phosphorylation, at least on your Western blot. Again, that asks, at least to me, for an explanation. It's on page 128.
Yes. Thank you, very learned opponent. Thanks again for the nice question. Indeed, we do not see a high level of change in the phosphorylation of GSK3 beta. However, I have to say that both bands are very faint, and mild differences might not be picked up like this, and we should then repeat a Western only for the LGR5 stem cells with probably a more sensitive detection solution where we could pick up differences.
Yeah, so, I don't want to pick points on technical details only, but the reason is that I spent 2 years working together with Clevers to show actually no indication of a connection between PKB, AKT, GSK3 regulation and the regulation of beta-catenin stability. And you basically take a review paper as saying, yes, there is a connection. So did you investigate that any further in any detail? Or did I spend my 2 years in vain? And did I do the wrong experiment?
Highly learned opponent, no. However, when dealing with Paneth cells and LGR5s, we do not know if the same pathways you observed previously in your work will work in the same way in these cells. For example, the same happens with the Hippo pathway. Yep. The Hippo pathway works completely differently in Paneth cells from LGR5 cells.
Yeah. Okay, thank you. Is there? So then a last question is basically asking whether there is a connection between your findings with the phospholipase and this chapter, because with the phospholipase you say after inflammation the Paneth cells need to remain Paneth cells, and in this chapter my interpretation is no, they need to de-differentiate and lose their Paneth function. So how do these two chapters relate, either philosophically or maybe mechanistically?
Highly learned opponent. We did not go into the details of which Paneth cells do differentiate. However, if you would allow me to speculate, sure. I have a theory about it. I believe that, as I was already hinting before, the younger and just mature Paneth cells are the ones who will be more prone to de-differentiate via lateral inhibition. And the ones who are already residing in the crypts 80, 90 days will be the one who will stay as Paneth cells, receive the phospholipase, and make the other Paneth cells de-differentiate.
So it's like real life. Older people don't change anymore, younger people do. Thank you for your answer. I give the word back to the rector.
Thank you, Professor Burgering. And then the opposition will be continued by Professor van Leeuwen. Professor van Leeuwen is Professor of Calcium and Bone Metabolism at Erasmus University. Professor van Leeuwen, the floor is yours.
Thank you very much, Rector. Dear candidate, first of all, congratulations with your thesis and also your supervisors. I want to include in that. I learned a lot from reading your thesis, a lot about Paneth cells, stem cells, etc. But you left me also puzzled at some point, so I want you to take me through it so that I properly understand what I've read. I'm not going to ask any question about Western blots. So, I want to continue on chapter 5 and proposition 4, that I perfectly understand what you mean there. In proposition 4, what you state is that the Paneth cells, in general, the Paneth cells are important for the control of the stem cells. That's correct, isn't it?
Yes.
Yeah, okay. No, that's good. I have read it. That was a good answer. And then in acute inflammation, the stem cells are lost. Is that correct? That's what you state in proposition 4?
Yes.
Yeah. And where can I exactly, you should help me, where can I see that in chapter 5? The more so, as you state on page 96, stem cell function of residual LGR5 cells is unaffected. And as I read in that sentence, that's based on the number of organoids formed. Mm-hmm. So I thought, did the stem cell number change? Or what happened? Because the number of organoids you see is not affected. So where can I see that the stem cells have disappeared?
Highly learned opponent, thank you for your question. Indeed, the few stem cells that remain in the lining of the gut do not change the clonogenic potential. However, if we fix the tissue and we look at immunofluorescence staining with specific LGF5 markers such as olfactomatin 4, we see a strong reduction of this marker.
But wouldn't you expect then that the number would be affected?
The number of stem
Organoids,
cells
I mean,
is
the number of organoids formed. Wouldn't you expect that to be changed then?
If you allow me to speculate, we did not elucidate that. However, I believe that there are two possible scenarios. One is that the residual number of LGF5 cells still keeps the same stem cell function as before. The other one is that the newly generated stem cells deriving from Paneth cells dedifferentiating have a very high clonogenic potential.
They compensate for the loss of the stem.
Yes.
Okay. But then coming back to the first statement that Paneth cells are important for the control of stem cells, how are these dedifferentiated Paneth cells then controlled? Which cells, the Paneth cells have been dedifferentiated. So are they then real stem cells, or have they an intrinsic machinery that they don't need a supportive cell?
By RNA-seq analysis, we see that there's no change in secreted growth factors or Notch signals. However, we cannot exclude that since not all Paneth cells dedifferentiate, the remaining ones still act as niche cells for the ones who are dedifferentiating.
Okay, and then I think that relates to the previous question about the age of the Paneth cells. I think that's what you answered before. So the older Paneth cells are still Paneth cells, whereas the younger ones dedifferentiate. And then if you isolate it, what's the impact of distance between the cells in this respect? I mean, is there a difference in distance between stem cells and young Paneth cells and the stem cells and the old Paneth cells? And what's the range that growth factors could travel to explain what you see?
Highly learned opponent, thank you for your question. As I stated before, we don't have tools to really understand which Paneth cells are 90 days old or 20 days old. As such, it becomes very difficult to localize them in the crypt.
That would be a real challenge for you now to find those tools.
To find a marker that would allow us to differentiate between mature Paneth cells, I believe that would be quite a breakthrough in the field.
May I ask you a question then, following up on this, if I look at figure, if I may still, do I have to, figure 3, panel C, following my questions, what kind of experimental condition do you think I would have added to panel C? Now maybe panel C, it's on page 125. What I was thinking, so maybe you can answer whether it's a stupid experiment, yes or no. Mm-hmm. Let's say the most right bar, that's the Paneth cells of the DSS. Mm-hmm. What about doing combination experiments with other normal Paneth cells? What would you expect to see there? Or is it, did you do it? And if so, what were the results? Or is it just you didn't do it because it's not a good idea?
It's a very good idea, first of all.
Thank you very much.
We did not do it, but if you allow me to speculate, I believe that some Paneth cells really do stay as niche cells, and as such, putting normal Paneth cells in the mix, it would improve even further organoid formation capacity.
So you think it would have been a nice addition to this panel to have that combination to prove really the role of things?
Yes.
Still some time?
Some time.
Then I want to move to the laboratory where you are now and their research topic and circadian rhythm. So at what time of the day did you perform your experiments here in Rotterdam? And did you perform them all at the same time point of the day? And do you expect it might have influenced your results if you would have done that, yes or no?
Thank you for your question. Most of the experiments were performed in the late morning. I do believe that for some of the findings, namely chapter 5 and chapter 3, this would not have an effect. However, I believe that the metabolism of cells would be different depending on the time point of the day. And I do strongly believe that that will change throughout the day.
So now in your new position, you will do it all at the same time point of the day. Is that correct? Thank you very much.
I thank you, Professor van Leeuwen, for your opposition. The opposition will be continued by Professor Griepnau. Professor Griepnau is Professor of Epigenetics at Erasmus University, and it is a pleasure to give you the floor, Professor Griepnau.
Dank u wel, meneer de rector. Dear candidate, I really read your thesis with great interest, and my compliments. It's a beautiful thesis, and as you know, we've developed, let's say, a recent interest in the intestine, and so we even sat together several times to think over experiments. And as you know, my interests are stem cells, and especially embryonic stem cells. But here, so while reading chapter 5, my first question is, so yes, I do believe that these Paneth cells play an important role in regeneration, but how is this in homeostasis? Do they also play a role in homeostasis? As stem cells, as generating stem cells?
Highly learned opponent. I do believe that fully mature, terminally differentiated Paneth cells do not play a role as stem cells in homeostasis.
But the young kids on the block?
However, secretory precursors are known to be slow dividing quiescent stem cells. As such, I do believe that the very early precursors of Paneth cells will contribute to regeneration.
Okay. So I think there are several, and you mentioned this in your introduction, I think even in the chapter, that there are also other stem cells, let's say, or other precursors of stem cells, like endocrine cells, but even enterocytes or early enterocytes that can do the job, let's say. Yeah? So if we think about these different experiments, here it was DSS, in other experiments it was gamma irradiation. So what makes the difference? So why in one experiment these cells show up and other experiments other cells? Or will all of them do this?
Thank you for your question. I believe that all of them do it. However, we are able to pick up one at a time because of the genetic mouse model that we use. So if we would use a specific Paneth cell tree, we would pick up only tracings from Paneth cells, and specific for enterocytes, only for enterocytes, and so on. But looking at tracing percentage, we do see that the total amount of regeneration cannot amount only for one cell type.
Okay. No, that's... But in homeostasis, if we go back in homeostasis, then yes, these young Paneth cells, they may do it, but do you also think that these endocrine cells and the young enterocytes may still do that? Or if we think about, let's say, the Cribb structure, is that a possibility or not?
I do believe that only in homeostasis, only LGR5 stem cells and secretory precursors, which are quiescent, will contribute to the population residing in the intestine.
Yeah. Then, so if you think about, let's say, this trans-differentiation event, and so as you know, we work a lot with iPS cells, and for us, the rule is that a cell needs to divide, and otherwise you cannot trans-differentiate or reprogram it to a different cell type. And so this example is completely different. And so the idea is that a cell needs to divide in order to dilute, let's say, its chromatin environment, its epigenetic memory. So how do you think that works here?
Highly learned opponent. Thank you for your question again. And I do believe that in this case, epigenetic marks will be wiped out. However, we did not elucidate this, and I'm just speculating about it. I do as well believe that this is not really a trans-differentiation, but rather a de-differentiation, meaning that Pange cells do not change their suit and become LGR5 cells, but they do start first upregulating transcripts from LGR5 cells, and then by asymmetrically apportioning this RNA, they will generate an LGR5 cell.
Yeah, I agree. I
think that would be a good idea, Professor.
I think I have to give the word back to the rector. Yeah? Thank you for your answers.
Thank you very much, Professor Griep. Now, and then will the opposition be continued by Professor Verijzer? Professor Verijzer is Professor of Biochemistry at Erasmus University Rotterdam. Professor Verijzer, please.
Thank you, Rector. First, dear candidates, first I want to congratulate you with the completion of an absolutely beautiful thesis. I've often glanced in the lab, so I know how much work it was, and I think it's really wonderful. So, questions. So I was wondering, I want to continue with this theme of de-differentiation of the Paneth cells. And I was wondering whether you could link that to Vogelstein's hypothesis that the incidence of cancer is closely linked to the number of stem cell differentiations that occur. So, for example, in inflammatory bowel disease and the risk of cancer. How would you place those things together, those different processes?
Highly learned opponent, thank you for your question. And well, it's a very interesting question. And if you would allow me to speculate about it, I would like to give my own opinion about that. The Vogelstein paper of 2015 postulates that indeed the mutations, and just by more mutations and cells dividing, people are susceptible to tumors. However, environmental factors such as inflammation, as you mentioned, impact greatly the rate of division and then, of course, mutations. And specifically addressing your questions about de-differentiating Pange cells, I believe that many of these so-called facultative stem cells, such as, for example, early enterocytes who will de-differentiate, or even Pange cells, are now dividing cells capable of acquiring mutation. And what I would do to address this study is I will mutate known genes in the adenoma to carcinoma sequence and then calculate the so-called lambda. And the lambda is the percentage at which cells are able to replace other cells in the crypt. And then we could calculate a mutated APC enterocytes who de-differentiate or a mutated APC Pange cells, in how much time will it take over the crypt and form an adenoma. That is my opinion about it.
Well, I couldn't have formulated it better myself. It's beautiful. So now to the environment, and the epigenome has been mentioned. So do you think diet, in what ways do you think, can you come up with, I'm a biochemist, can you come up with a potential biochemical pathways that might affect these processes, depending on what you eat, for example? For example, the amount of fiber in your diet.
Thank you for your question. Diet and stemness and colon cancer are being investigated very thoroughly, and indeed, diet with low fibers, for example, do promote colon cancer, and I believe that that will affect the stem cell niche. Vitamin D is another factor that influences as well all of these processes. And more recent publications from the ILMAS group in Boston indicate that high-fat diet endows other cells with stem cell properties via the PPAR-gamma pathway. However, I do not exclude that the niche, meaning the panel cells, will also be influenced by dietary effects, and that they will also respond to those.
So, for example, the bacterial digestion of dietary fibers produces enormous amounts of butyrate. So you get very high local concentration of butyrate, which of course is a very strong inhibitor of histone deacetylation. And for example, in the hematopoietic system, that can really influence cell differentiation. So really cranking up on a very global level, the level of acetylation of the genome. Do you think that could affect susceptibility to dedifferentiation or not?
Absolutely. There is actually work on this by the group of Tadeusz Stappenbeck. And what the group elucidated is that butyrate secreted by bacteria inhibits LGR5 proliferation. As such, I do believe that that will also have a negative effect on the differentiation of panel cells. However, this has not been tested.
Okay. Are we time-wise? Is there?
Time-wise, I think your time is over, Professor.
Okay. Thank you very much for your answers. Thank you.
Thank you very much, Professor Verrijzen, for your opposition. It is a pleasure to give the floor to Professor Looyenga, and Professor Looyenga is Professor of Translational Patho-Oncology at Erasmus University. Professor Looyenga, the floor is yours, if it works. Your building.
Thank you, Mr. Rector. I have to shout a little bit because the microphone is stuck. For sure, I want to congratulate you first with the result of a lot of work, and also include your promoter with this end result. And of course, I have a number of questions to discuss with you. Reading the propositions, I was a little bit surprised, but because my idea was always that you need 5 propositions related to the for the thesis, then 5 related not to the thesis but defendable in a scientific way, and then 1 free, more general proposition. Reading your proposition, I think you forgot category 2, but by reading them, I really saw the imprint of your promoter, and that's also not that unexpected, because I think you met already when you were 16. So that's enough about the propositions, then going to the results, and again also related to chapter 5. And you know, of course, that I'm also interested in the KIT ligand, stem cell factor, and looking at your illustration figure 1, my first question is, and it was already a little bit mentioned by you in answering the question of Professor Bergering, what is the origin of KIT ligand in the system you're looking at? And you're analyzing the protein by immuno and the messenger RNA by in situ. So what is in your view, because everywhere I read you're a little bit vague and sometimes a little bit more specific in other places, what's the origin? Where does it come from?
Highly learned opponent, thank you for your question. I believe that the KIT ligand in our setting has a multiplicity of origin. As shown by the RNA scope, there is KIT ligand being secreted by the epithelium, but I believe as well that inflammatory cells who are known to secrete the KIT ligand will invade the mucosa and secrete KIT ligand under inflammation, and as well as a subset of stromal cells will secrete the KIT ligand.
And I checked again this morning, both in humans and in mouse, there are various variants, including the membrane-bound and the soluble, different in usage of exons. What do you think is here the most profound variant doing the job? You did not check it, what I've seen so far, but what do you think?
I believe that the most profound variant is a secreted form by the immune cells under inflammation. However, some epithelial cells we analyzed by qPCR do have the non-secreted version.
So it's both, but possibly heterogeneous.
It's possibly heterogeneous, yes.
Because Joe Nadeau from the US also reported that there is a major difference between the membrane-bound and soluble related to susceptibility to cancer in mice. So that's also interesting in this context. So it's heterogeneous, but is it now really true what you answered in the beginning of this defense, that it's changing to an autocrine system in vitro?
Highly learned opponent. Thank you for your question. I do believe that in vitro this becomes an autocrine system because we are sorting pure Paneth cells. And we did examine that Paneth cells do not have the KIT ligand. If we would be plating multiple cell populations, I would not be able to exclude this. However, since we are purifying Paneth cells by flow cytometry, I think I can exclude that this is that this is not an autocrine system.
And related to that, as you know, I have a specific topic of research. There are some similarities with the work you've done related to the supportive cell and the stem cell. So do you think that this system you are investigating, and you are saying it's more de-differentiation of the Paneth cells, can also occur in other organs, and which organs specifically then?
Thank you for your question. I do believe that this occurs in other organs. This has been proven in stomach epithelium. It has as well been proven in pancreas and liver as well.
And what do you think about the testis?
I do see many similarities with Sertoli cells and Paneth cells, and I do believe that Sertoli cells could overlap Paneth cells in function. Interestingly, Sertoli cells express high levels of phospholipase 10, which has been described here as a tumor modifier.
And SOX9 as well. And if I'm allowed to add one particular short...
Last short question,
Yes,
Professor
very-
Loinga, and the short answer if possible.
Related to the germ cell cancers, there has been done a lot of genome-wide association studies. A number of risk SNPs have been identified. The majority of those link to the KIT ligand and downstream pathway. And my simple question is that also found in IBD? Is there a link with variations in the genome and this disease?
Thank you for your question. There are more than 300 SNPs identified in IBD. However, to my knowledge, they do not link with the c-KIT ligand pathway.
Thank you for your answers, and I return the word back to the director.
Thank you, Professor Loinga, for your opposition. And then I give the floor to Dr. Cupido. Dr. Cupido is Assistant Professor of Hematology at Erasmus University. Professor Cupido.
Dear candidate, I too want to start by congratulating you with this beautiful piece of work. I very much enjoyed reading it. I really like the fact that you start off by introducing a technique that then is reiterated throughout your chapters and used to make the points. So congratulations on that. So I want to start with a question that sort of came up after listening to your presentation. So where you stated that the nature and the function of these LGR5 stem cells is really to replenish all these epithelial cells that we lose every day. So, but then you come in with all these, with this DSS model, which basically damages epithelium, putting the system into stress and really requires at that point massive regeneration. So it's the normal situation amplified. But then what happens is that all these stem cells die. So that seems to make little sense. So why do you think that these stem cells disappear at a time when they are most needed?
Very learned opponent. Thank you for your question. I do believe the reason why stem cells disappear at the moment when they are most needed is because evolutionarily there is one genome that we do want to protect, and that is a genome of our stem cells. So to avoid mutations, I believe that an evolutionarily way to protect the genome of stem cells is to hide in differentiated cells the source of regeneration under very acute inflammation or very heavy tissue injury.
Okay, that's an interesting hypothesis. So do you have any ideas on what makes the stem cells die? So you sort of call it inflammation in the thesis, but what is that inflammatory signal that makes that these stem cells die?
Very learned opponent. We did not elucidate this. However, if I may speculate about this, in chapter 4 we described that LGF5 stem cells rely on oxidative phosphorylation to harvest their energy. And this generates a large amount of ROS. And I believe that in inflammation, a lot of ROS is already released by immune cells. Indeed, alphamine is a SNP in IBD, and that is because of less ROS production. And I believe that too much ROS is just not good for the stem cells, and they die because of that.
So do you think that it involves actual DNA damage? So to come back to your previous hypothesis on safeguarding the genome in post-mitotic cells, that it is the process of DNA damage in the stem cells that is the trigger for them to die? Perhaps induced by ROS or others?
If we look at the historic perspective of how inflammation has been shown to work, there is never a genotoxic effect from inflammation directly on the cells. However, we cannot exclude that that might be the case in this very specific setting.
Okay. So I have another question which relates to the phospholipases that you describe. So what I found very intriguing to read is that you state that these phospholipases are actually secreted into the intestinal lumen. And to me, sort of as maybe the ignorant reader, I would have expected that they would have been secreted at the basolateral site into the tissue where most of the cells that they should act on are, where Bannet cells have lots of receptors. So why do you think that they're secreted into the lumen and not into the tissue?
Very learned opponent. Thank you for your question. Phospholipases are, before being genetic modifiers and before being stem cell niche modifiers, known for their antimicrobial function. To elicit this antimicrobial function, they need to be in the lumen, in contact with the bacterial flora, where the bacterial flora reside. However, there is still the possibility that part of this phospholipase might be secreted laterally to other cells.
And you also looked at the receptor for the phospholipases. And as far as I know, this is a receptor that is still it's a bit enigmatic, right? So it doesn't really have a big intercellular domain, and we really don't know how it signals. So what I get from literature is that one of the prevailing hypotheses is that it probably needs other receptors or amplifies other signals. So do you, and again, this comes back to the basolateral versus apical side, because these C-type lectins can often sort of associate with TLRs, and most of those are on the basolateral side. So do you have sort of any indications from your work that, and based on the signaling pathways, how this receptor really transmits signals? Does it need another receptor, or is this just a standalone?
Thank you for your question. From the work that we have done, what we have seen is just that phospholipase, by binding directly to the receptor, triggers the activation of the cytosolic phospholipase 4-alpha. However, we did not study if the specific receptor needed other receptors in combination to trigger the phospholipase activity. But what can be desumed from the work is that the absence of this receptor does not elicit the regenerative response of phospholipases. As such, it is required the receptor to bring the phospholipase in.
Mr. Candidate, the time allowed for the defense of your thesis is over, and the committee will withdraw to deliberate. Please wait here until we return. I adjourn the meeting.
Hou je wel op tijd. 15 minuten werk. Dus. Vind je goed? Ja, ik denk wel. Niks meer te zeggen? I reopen the meeting. Mr. Candidate, the doctoral board of Erasmus University, represented by me, has considered your thesis. Having heard your defense and the advice of the committee, I grant you the title of doctor. And I authorize Professor Vodde to bestow this degree upon you in the customary manner.
It is with great pleasure that I undertake the task the Rector Magnificus has allotted me. By the authority invested by law and according to the decision of the doctorate board of this university, I hereby confer upon you Matthias Herwe, the doctoral degree. As a testimony and proof hereof, I present you with the diploma signed by the Rector Magnificus, promoter, and other members of this committee, and endorsed with the seal of the university. No, wait. No pictures? No. Having performed this task, may I take the opportunity to be the first to address you as a doctor and to congratulate you on your achievement. Dr. Herwe, dear and carissimo Matt, as I and everybody else calls you. Today, Wednesday, September 19, 2018, is the very last day of a journey that started in the spring-summer of 2006, in fact, when a half German, half Sardinian, almost 2 meters tall and forward-hunched high school student, accompanied by his very apprehensive parents, came to visit me in my parents' home in Italy to ask advice on the university career to follow in order to become a scientist. And the rest, as they say, is history, which, in a nutshell, I would like to summarize here in 2 or 3 milestones. First of all, number 1, you went on to study biological science in Pavia as a bachelor. Then you succeeded in that, and you tried to enroll into several master degree programs, both here at the Erasmus MC, in Heidelberg, and elsewhere, and then experienced your very first scientific disappointment, when your passion and talent were not recognized by my own Dutch colleagues, something that I turned to the Dean of the faculty, probably the newly formed Research Council of the Erasmus MC should focus its attention on. Number 3, finally, upon completion of your Master of Science diploma in Pavia in the laboratory of a fellow of mine, Professor Fiorenzo Peverali, I knew I finally had a chance to give a PhD job to someone truly in love with science and prepared to blood, sweat, and tears in order to achieve his scientific goals. The many nights you spent in the lab during your first years are just a small testimony of that working attitude, even though I'm not so sure that quantity and quality goes together when it comes to working hours. Having said that, there is always a but. When I gave you the PhD job, I was also aware that you were going to be the worst nightmare and pain in the neck I've ever had in my lab, which in fact turned out to be a huge underestimation on my side. So, jokes aside, the Italian expression genio and sregolatezza, which is translated for the non-Italian among us as genius and recklessness, is particularly fitting and appropriate for you. Your scientific curiosity, passion, creativity, and extended knowledge come together with a huge allergy for whatever is discipline, order, and structure. I still have a regular nightmarish vision of your unintelligible, even for yourself, logbooks, the meter-high piles of all kinds of papers, coffee cups, beer bottles, and other unidentified objects on your desk, and of your bench, which looks more like a battlefield than anything else. These are aspects of your being a scientist on which you'll have to work very hard in the future, and especially by the time you are a PI yourself. It should also be said that your PhD, as it's almost always the case, is the result of a group's effort. And you were by all means the right person in the right time, in the right place. Your two para-nymph, I didn't know how to translate the word into English, so I will go for bridesmaids. Patrick and Mark have been pivotal for your own development as a person and for the successful implementation of the experiments in the laboratory. But we should also not forget Danny, Miriam, Rosalie, and many others in the labs who contributed to your successes. Likewise, you had the fortune to collaborate with many great colleagues and laboratories, both in the Netherlands and abroad. Maria, who is here today, Maria Rodriguez-Coleman, Professor Burgerding, whom I thank for being in the commission, Bob Cormier in Duluth, Minnesota, the city where Bob Dylan was born and where Matthias also performed some experiment. Gérard Lambeau in Nice, France, where you also performed some experiment for your thesis. Also on your behalf, I would like to express my sincere gratitude to all those who contributed as collaborators to your PhD work. Going back to your early university days, the legend goes that one day while you were still at the Collegio Ghisleri. This is a residence for university students in Pavia, where Mattia resided and so on. One day, Professor Fraccaro met you while you were just sitting on the floor, I don't know exactly the details, reading eagerly the last issue of Nature. And it sounds like Professor Fraccaro said, one day your own paper will be published in Nature and so on. Well, his prophecy was certainly, I lost my pages. His prophecy became a reality, and today's PhD is the cherry on the pie on this long, winding and bumpy road that led you to become the postdoctoral fellow in a very prestigious laboratory led by Professor Pekka Kataisto, whom I also want to personally and most wholeheartedly thank for his presence today. Dear Matt, dear Dr. Schewe, I finally come to the end of my laudatio, but I cannot do that without mentioning and acknowledging the three most important persons in your life, and I'm not one of those. As you wrote in the acknowledgements, your wonderful girlfriend, Lan, and your lovely parents, Thomas and Annalisa, whom, if the rector allows me, I would like to briefly address in our own mother tongue. Carissimi Annalisa e Thomas, vi sarò sempre grato per avermi portato Mattias, in quel lontano giorno del 2006, a casa di Mirella al Poetto. Oggi, 13 anni dopo, Matt, insieme a voi e grazie a voi, ha raggiunto un traguardo importantissimo, sia dal punto di vista professionale che da quello umano e personale. E so bene quanto ne siate fieri e orgogliosi. E avete tutte le ragioni di esserlo. E adesso che le formalità accademiche sono terminate, potete rilassarvi, ridere, piangere, essere felici e festeggiare insieme ai colleghi e agli amici di Matt questa giornata molto particolare e spero per voi indimenticabile. Geachte Dr. Schewe, het gaat je goed. En ik geef het woord terug aan de rector magnificus.
Dr. Schewe, I also have the pleasure, on behalf of the doctoral board of the university, to congratulate you on having obtained your doctorate degree. And I formally declare this meeting closed.